So good morning, everyone. Thanks to the organizing committee for inviting me for the short talk in this section. So I am Lhais Caldas. I just finished my PhD in the Federal University of São Paulo, Brazil. And this talk is entitled Biosynthetic Gene Clusters of Geminopilus imperialis, Secondary Metabolites with emphasis on polyketate synthesis. So this is an integration of metabolomic and genomic tools. So first of all, I would like to start, I would like to start giving an overview about Basidium mycota, which is also known as mushrooms. So it is the second largest phylum of fungi in the world, with approximately 1,600 genus cataloged and an unknown number of secondary metabolites being produced by them. So in Brazil, we have around 2,700 species cataloged. They are mostly from Agaricalis, and they are mostly also from the Atlantic rain forest, so known as Mata Atlântica biome. So one important thing to highlight here is that less than 10% of this original biome is still alive nowadays. So together with all of these vegetation and organisms, we are also losing a lot of chemodiversity and maybe drug prototypes. What we know from up until now about basidiomycetes chemodiversity is that they produce a large variety of small molecules, also known as secondary metabolites, and that they have around 130 pharmacological properties recorded. Most of them are anti-tumor, antimicrobial, antioxidants, and many other pharmacological properties. But our interest is mainly on Geminopilus imperialis, which is one native species from the Atlantic rainforest in Brazil that we collected a few years ago. So then we decided to access the metabolomic profile and also the genomic profile of this mushroom. So our main goal here is to make use of omics sciences to access the full biosynthetic potential of these species and identify novel fungal metabolites, especially bioactive metabolites. So now we are focusing on antibiotic potential. So for this, we aim to direplicate these extracts through metabolomics, especially mass spec, tandem mass spec, identify gene clusters related to the biosynthesis of the annotated molecules through a heterobiosynthetic approach. And finally, heterologous express these BGCs, these genes of potential novel bioactive metabolites. To achieve these goals, we are focusing mainly on genomics and metabolomics. So we started by collecting and cultivating these species. Then we prepare a few extracts, especially liquid extraction, to get more polar and non-polar extracts. Then we ran metabolomics analysis with LCMS-MS, and finally, with the aid of GNPS, we dereplicated and created molecular networking to annotate metabolites. Concerning the genomics approach, then we sequenced the genome of Geminopilus imperialis, assembled the genome, and then we use conscious mesh to do the BGC prediction. So we wanted to focus on the biosynthetic gene clusters. Also, we decided to use a phylogenetic approach to link metabolites to our own sequences, to our own genes, and then comparative genomics to select potential novel BGCs, potential novel metabolite backbones to heterologous express in filamentous fungi, which in this case is Aspergillus oryzae. And then we finally screened for the metabolite production. So to start with the metabolomics approach, with the aid of GNPS, we got this molecular networking, and then what we could see is that these species produces mostly terpenoids, stereopyrones, steroids, and oligoisoprenoids. The major compounds are actually geminopilins, which is this oligoisoprenoid, and the stereopyrones histidine and bisnaryonganin. At the beginning, we didn't have any information about literature data about these species, so everything we knew about metabolomics was new. So then we decided to access the full biosynthetic potential of these species. So we wanted to compare BGCs conserved in related species. We also wanted to assign metabolites to their respective BGCs and using this heterobiosynthetic approach, and access possible cryptic biosynthetic pathways, and finally select possible novel BGCs for cloning. So then we used 3 related species, which are Geminophilus chrysophilus, Geminophilus genonius, and Gallerina marginata. So then we did the whole genome mining approach using this related species together with Geminophilus imperialis. And we use Gallerina marginata as the template for heterologous expression. So we use these 3 species and Geminophilus imperialis to do the biosynthetic gene cluster prediction. So we started by predicting the genes using Augustus, and then by predicting the biosynthetic gene clusters using Fungus mesh. So from this first approach, we can see that they all produce similar number of BGCs, with the exception of Geminophilus imperialis, Geminophilus genonius, and that they encode mostly for the production of terpenes, peptides, and polyketides. So based on our interest on polyketides, as we know that they can be very diverse in fungi, and also that they have this potential to be bioactive, especially antimicrobial. So then we decided to use a phylogenetic approach to link BGCs to metabolites. So then we use MIB fully characterized fungal synthesis together with our own sequences from the 4 species, the 3 related species, and Geminophilus imperialis. And then we could see 3 well-defined clades that we selected for further analysis. So we have 1 hybrid NRPS PKS cluster, 1 highly reduced PKS, and 1 non-reduced PKS cluster. So we decided to check to evaluate how conserved these BGCs were among the 4 related species. So then we ran comparative genomic analysis, and then we could see that they are all very similar to each other, which suggests that they probably encode for the production of similar metabolites or similar structural backbones. So we selected these sequences for heterologous expression, expecting that we would get polyketides, novel polyketides, bioactive polyketides at the end. So then we started the heterologous expression by preparing our plasmid vector with yeast, Saccharomyces cerevisiae in this case. Then we selected the true transformants to prepare our expression vector in E. coli, and then we finally selected the true transformants to transform into filamentous fungi, which in this case is Aspergillus oryzae. So we did a back-mediated transformation using Aspergillus oryzae. Then we isolated the colonies. We selected 3 times in different selected media, and then we screened for the metabolized production. So what we expected to see here was the production of polyketides, which probably would have the same backbone as the clades that we selected at the beginning of the genome mining, which are osporine and istrobutylene. But unfortunately, we couldn't see the production of these metabolites. But this is an ongoing project, so this is work in progress. We are still making a few adjustments in the transformation protocol in order to stimulate the production of polyketides by Aspergillus oryzae. But in general, what we can see from this project is that we could access the full biosynthetic potential of Geminophilus imperialis and also the related species, Geminophilus and Galerina genus. We also could see that Geminophilus imperialis, it produces 41 VGCs in total. They are mostly sesquiterpene syntases and PKSs. We are also working with sesquiterpene syntases. We could assign metabolites to their respective VGCs. This was what we were expecting at the beginning. Use this heterobiosynthetic approach. And as I said, this is a work in progress. So we are still improving the heterologous expression of the PKSs, and I hope that soon enough I will have polyketides to report. I would like to thank the founding agencies, my lab networking, and all of the collaborators, and also the organizing committee for inviting me for this section. Thank you.
Great, thanks, Lhais, for a great talk. We're going to take 2 questions. Satri.
Thank you for the talk. I have, can you hear me? Okay.
Yeah.
Can you elaborate more about this retrobiosynthetic approach? Like how you will link the metabolites with the BGCs using this phylogeny of the PKSs?
Yeah, we can do it in many different ways. In this case, we started by using metabolomics approach. But then it is also possible, but we try to link the metabolites that we already had, the annotations that we already have with the biosynthetic gene clusters, like manual creation. But it is also possible to make use of a few bioinformatic tools. Right now, for natural products, we have a few integration tools, which are NPO mix and NP linker. This is, it makes an integration more easily, so it assigns exact metabolites to their family cluster genes. So this is more accurate, in fact. But in this case, we wanted to evaluate if we were losing metabolites in our metabolomics approach. So we wanted to get to know the whole biosynthetic potential of this mushroom. So the goal was to know all of the biosynthetic gene clusters, and then we could take a step back and try to assign them to the metabolites. So a few things we still have to work on.